Hello and welcome back to the channel. Today we're going to be discussing eukaryotic cells, in particular the nucleus, mitochondria and chloroplasts. So let's jump right in. The nucleus is enclosed by a nuclear envelope, a double membrane that separates the nucleus from the cytoplasm. This envelope is vital. It protects the genetic material inside and regulates what enters and exits. For example, it allows essential molecules like RNA and certain proteins to pass through, while keeping harmful substances out. Now, the nuclear pores are embedded within the envelope. These are tiny but crucial structures. About 3,000 pores are typically present in a single nucleus. They act like selective gateways, permitting the transport of large molecules such as mRNA out of the nucleus. This export is critical for processes like protein synthesis, which happens outside the nucleus in the cytoplasm. Moving to the inside, the nucleus is filled with nucleoplasm, a jelly-like substance. This material provides structural support, helping to suspend and organize the internal components of the nucleus, like the nucleolus and chromosomes. Next, we have chromosomes. These are made up of protein-bound linear DNA. Think of chromosomes as the cell's genetic library. They carry out all the instructions needed for the cell's development, function, and reproduction. In humans, there are 46 chromosomes in each nucleus, containing thousands of genes that dictate everything about how our bodies work. Another key structure is the nucleolus, which is a dense, spherical region within the nucleus. Its primary function is to manufacture ribosomes, which are tiny molecular machines responsible for building proteins. The nucleolus assembles the components of ribosomes, which are then transported out of the nucleus to perform their role in the cytoplasm. Without the nucleolus, cells wouldn't be able to produce proteins, and they wouldn't survive. Now let's explore the functions of the nucleus. The first major role is the control center of the cell. The nucleus directs all the cell's activities by regulating gene expression. Genes, which are specific sequences of DNA, act as instructions for building proteins, and the nucleus ensures the right genes are expressed at the right times. For example, if a cell needs to produce a particular enzyme, the nucleus activates the gene responsible for that enzyme and facilitates the production process. The second function is that the nucleus stores the cell's genetic material. DNA within the chromosomes contains all the instructions needed for the cell's operation, growth, and replication. Importantly, this genetic material is carefully organized and protected within the nucleus to ensure it remains stable and intact. Any errors or damage to DNA can have severe consequences, potentially leading to diseases like cancer. Finally, the nucleus is essential for ribosome production, which happens in the nucleolus. Ribosomes are assembled here before being exported into the cytoplasm, where they play a critical role in translating genetic information into proteins. These proteins are used for everything from building cell structures to catalyzing biochemical reactions. So now let's take a closer look at the structure of the mitochondria, the powerhouse of the cell. Mitochondria are surrounded by a double membrane. The outer membrane acts as a boundary, enclosing the organelle and allowing selective entry and exit of molecules. The inner membrane, however, is where the real action happens. The inner membrane is highly folded to form structures called cristae. These folds are not just for show, they significantly increase the surface area available for metabolic reactions. This is crucial because on the cristae, the enzymes and proteins involved in respiration are located. These enzymes help drive the process of energy production, making the cristae vital for the mitochondria's function. Inside the inner membrane, we find the matrix, a jelly-like substance that makes up the rest of the mitochondria. The matrix is packed with important components, including proteins, lipids, ribosomes, and even mitochondrial DNA. This DNA is unique to mitochondria and allows them to produce some of their own proteins independently of the nucleus. This ability makes mitochondria semi-autonomous, meaning they can partially control their own function. So now let's talk about what mitochondria actually do. Their primary role is ATP production. During aerobic respiration, energy is extracted from glucose and other molecules, and this energy is stored in the form of ATP. ATP, or adenosine triphosphate, is often called the energy currency of the cell because it powers almost all cellular processes. Because of their vital role in energy production, mitochondria are especially abundant in cells with high energy demands, like muscle cells or neurons. For example, your heart muscle cells, which are constantly active, contain thousands of mitochondria to meet their energy needs. So to summarize, mitochondria are double membrane-bound organelles with an inner membrane folded into cristae to support efficient energy production. Their matrix contains the tools needed to produce proteins and sustain the organelle's semi-autonomous function. Their ability to generate ATP makes them indispensable for cell survival and activity. Okay, so let's take a closer look at chloroplasts, the organelles responsible for photosynthesis. Chloroplasts are surrounded by the chloroplast envelope, a double plasma membrane. This membrane controls what enters and leaves the chloroplast, ensuring the right conditions for photosynthesis. Inside the chloroplast, we find structures called grana. Grana are stacks of disc-shaped structures known as thylakoids, and these thylakoids contain the pigment chlorophyll, which is crucial for capturing light energy. The grana are the site of the first stage of photosynthesis, known as the light-dependent reaction. During this stage, light energy is converted into chemical energy in the form of ATP and NADPH. The stroma is the fluid-filled matrix that surrounds the grana. This is where the second stage of photosynthesis, the light-independent reaction, or the Calvin cycle, takes place. In this stage, the ATP and NADPH produced earlier are used to convert carbon dioxide into glucose, which the plant uses as an energy source. Now let's talk about how chloroplasts are adapted to perform photosynthesis so efficiently. First, the granule membranes have a large surface area. This maximizes the space available for chlorophyll, electron carriers, and enzymes involved in the light-dependent reactions. More surface area means more light can be absorbed and more reactions can take place. Next, the stroma contains all the enzymes needed for the light-independent reactions. These enzymes work together to convert carbon dioxide into sugars, making the stroma an ideal environment for this process. Finally, chloroplasts contain their own DNA and ribosomes, allowing them to quickly produce the proteins and enzymes they need for the photosynthesis. This semi-autonomous nature helps chloroplasts adapt to the cell's energy requirements. So, thank you very much for taking the time to watch this video on the eukaryotic cell structure. In this video, we discussed the nucleus, mitochondria, and chloroplasts. Make sure you come back for part 2, where we'll discuss all the other organelles that are found within eukaryotic cells. Make sure you subscribe, like, and comment. Share this video with friends if you found it useful, and also feel free to check out any other videos on the channel.